So yeah, my topic today is an out-of-body experience. What I'm going to try to explain to you today is what researchers do when they get cells from the body. What options do we have, and how we are making this better. So I have to begin by saying, what is cell culture? Um, Essentially, it is the isolation of cells from the body. And we grow these cells outside the body, typically in flasks. That's the go-to standard as of now. And this is under very controlled measures, very controlled conditions, really. And it's obviously outside the natural environment. They're not in the body anymore. We isolate them, and we try to grow them, expand them, and use them for research. But we have many options here, and we have 2D cell culture and 3D cell culture. Now, it's essentially the same coin, but two different sides of it, right? So relative to each other, which is, let's talk about 2D cell culture first. And it is relatively less complex compared to 3D, given that it's a flat surface. It's only two-dimensional. Also, this really is an inaccurate representation of what happens in the body. We are not 2D creatures, really. We're 3D, made of different tissues. And, well, 2D cell culture is typically done on plastic. And as you probably know, many plastics we use in the lab are hard substances. Now, that doesn't sound like the body in many ways. When we look at 3D cell culture, it is certainly a lot more complex compared to 2D cell culture, and also represents a lot of the dynamics that go on in the body in terms of diffusion, in terms of how things flow through the body. We are 3D structures, right? And these often require biomaterials. So it's combining biomaterials and cells to create an environment that would best suit them as though they're still inside the body. So they're kind of having an out-of-body experience here. And these are often soft materials, right? Given that a lot of the tissues in our body are soft, or at least start off soft, and then get mineralized, for example, like bone. So we work with soft materials here. Let me introduce you to the project we work with. It's called 3D Life. And the idea here is, can we really make cells feel at home outside the body? Can we create a sort of substance or a biomaterial where we can maintain cells rather than having them change and adapt to these hard surfaces? So I'm going to walk you through this diagram here. When you look at typical cell isolation from the body, we isolate cells, grow them in 2D, and as they get very comfortable, they tend to adapt to their microenvironment. They change. They get used to this hard substance rather than being used to this soft substance that's around in the body. So what we try to address here with 3D Life is, when cells are isolated and we have them in culture in these gels that we use, could we then keep them as they are, as they were in the body? And to do so, we evaluate whether the cells are expressing the same genes that they would in the body. Are they thinking the same? Are they producing the same proteins as they would in the body? The idea here is to create essentially a library of materials that you could use for different cell types. Now we know cells are different in the body. They adapt in different microenvironments. So could we have a set of gels that can be used for example, a different set of gels for bone cells, a different set of gels for lung cells, and such. So that's really what we're addressing in 3D Life project. So the question, of course, appears, what cells do we use for this? And what biomaterials are we interested in? And there are very many options of both cells and biomaterials, right? So the cells we use, as Maria mentioned in her talk, the word fibroblasts. So I'm going to introduce you for a second what fibroblasts actually are. Now, as the name suggests, fibro, given that it produces fibers, and a lot of these fibers are actually collagen fibers, for example. And these cells are mainly responsible for putting cells together to form tissue. They kind of are the glue, in a way, to bring cells together to form a tissue. And where are they found? Honestly, pretty much in every organ. And one such organ is skin. So we have cells present in, fibroblasts present in the dermis, which produces collagen, and provides this kind of matrix for the epidermis to form on top. And why is it interesting, or why are we even using fibroblasts? Well, firstly, as I mentioned, they are abundant all across the body. But at the same time, we know a lot about it. We've studied these for many, many years. And they're very easy to culture. They don't really require very unique conditions. But at the same time, as I mentioned, fibroblasts are also present in other tissues, such as lung, the gingiva, which is the lining of your mouth, around lymph nodes, for example. But one more cell type we're interested in is also stem cells. Could we maintain stemness, for example? So the idea is we isolate, or we have cells that are isolated from lungs, the gingiva, and the lymph, which are all still fibroblast cells. So can we maintain their natural microenvironment in a biomaterial, for example? And that's what the goal of 3Life has been. Now that you know the cells, what material do we use? To put it very plainly, we work with chemically modified alginate from brown seaweed. Now, if you put on some goggles and dump your head into the water in the Trondheim fjord, you might see something like this, all right? But we process this a bit. And one of the substances we're interested from this brown seaweed is called alginate. So I'm not going to get into the depth of it, but alginate essentially is a polysaccharide, a sugar molecule essentially. That's made up of these two blocks, which we refer to as G blocks and M blocks. Now, what the unique thing about alginate is that we can form gels using calcium, for example, as an agent. But there also are problems with alginate. It's quite inert for cells. So we have to modify this in a way that cells actually like them. And the two modifications we're interested in, or mainly two, is adding sulfation and adding a molecule that will allow cells to adhere to them. So kind of making cells feel more at home. When we culture these cells from different tissues into the gels, so over here you can see what we call a live dead image. The cells you see that are in green are live, and the one in red are dead. So as you can see, majority of the cells survive in our gels, both at day 1 and at day 7. That's a good sign. What do we do with this? We can then extract a huge amount of data where we can see what the cells are expressing, what are they thinking, how do they feel essentially on a macro level. And are they close to how they were when they were in the body, having taken them out and putting them in these gels? How much have they really changed? But it doesn't stop there, right? Now with this alginate, we can do a lot more things. We can produce beads, for example. We can control the structure and guide these cells to orient themselves like they were in the tissue. So for example, you see here these beads of alginate that we've then colonized with fibroblast cells again. And they tend to surround the structure. Now, if you have a basic understanding of anatomy, this might remind you of lungs. So you see the alveolar structure in lungs look kind of like this. So we're onto something here now. We're trying to replicate what happens in the body structurally as well. So now I have one application, but so what? What can we really do with this knowledge? And there are a few things that the 3Life brings about. Well, firstly, the fact that we use marine-based biomaterials, which directly feeds into the Norwegian economy in many ways. And it's also very abundant. But also, as you've seen before, we're trying to create these model systems that mimic how tissue actually is, how it's structurally in the body. Overall, if we do create these models and they function as expected, we would technically be able to use them and reduce animal studies. We can test pharmaceuticals and such. And also, we can actually translate this to other mammals, which you'll see in a second. So I'm going to tell you a short tale about a whale very quickly. So this is, no, I'm not going to give him a name. It'll be too weird. All right, so we work in a project that's a cross-collaborative project with the University of Bergen, with SINTEF, with UC Berkeley, and University of Tromsø, where we actually are trying to create these similar environments for the whales. So what we do is here again, which you're familiar with, fibroblasts from the skin, isolated and cultured. Now this, mind you, is a 2D picture, just to show you how big these cells actually are. So right, so the whole concept here is, can we actually replicate whale tissue in order to evaluate how environmental toxicants affect these organisms? So could we create a model system that would replicate how these cells are affected? And there's a lot more we can do, right? But I'm going to stop right there. And I'd like to thank everyone who's worked in this project. Yeah, thank you.